We're really excited to have Professor Krishna Shrinivas here to talk to us today, so I won't take up any more of your time.
Thank you, Alistair. Thanks, everyone, for having me. I'm excited to see more people here. This is an awesome space, and I hope you guys stick around for later in the day to hear about how you could come and use this awesome resource. I'm a little late on time, so I don't know what the time is. It's okay if we don't get to the end of the talk. I have a bunch of introduction on a particular class of problems we think about. It's a little light on specific math, but there's a blackboard, so if you have questions, just ask me. And just stop me when I'm talking. It's easier to go through stuff if we are all on the same page rather than getting till the end. A very brief introduction. My name is Krishna. I am an assistant professor in chemical and biological engineering at Northwestern. I started last year. I have a small group. They're all here. Most of them are here. Second row. Let's see whether this slide advances. Okay, that's us. And sorry for the blurry picture. So today we're going to be talking about work from Jackson and Neha, who are both here, as well as another grad student who works with me from Harvard, Ryan Kruger. And we are broadly thinking about building theoretical and mathematical approaches to study biological processes. Okay, so the kind of overarching interest, at least for me particularly, and for folks in the lab, is like asking the question, how does life run in the crowded environment that's a cell? Do you want me to change anything? No, okay. That is a physicist's caricature of a cell. It has stuff in it, like the genome and mitochondria. But if you look at the inside of the cell, it's not quite what we might think of when we think about, interacting systems or chemical systems. It's an incredibly complex environment. This is a simulation of a bacterial cytoplasm from Michael Faig's group. It's incredibly crowded. There are about 1 billion to 10 billion different biological molecules, 1 million different species that are all interacting, reacting in this non-equilibrium environment. And so our lab just asked, like, how do we get the process of life to run reliably in this dynamic, fuzzy, stochastic environment? Okay, so there's one really good paradigm to think about how order and structure emerges in biology. And this is just an introduction of something that we all know. Okay, so it's this really successful paradigm that's really defined the last 50 years of molecular biology. It's the idea that the sequence of a biological molecule, like a protein, natively encodes for its stable, folded, three-dimensional structure. And that structure can be interrogated and studied to understand how it encodes for a specific catalytic function or a function to build a larger macromolecular complex or a cellular wall, things like this. Innumerous examples of these particular sequence structure function relationships. They define most of the protein biology we know today, and it's been successfully used to understand how proteins work in synthetic and biological contexts. Okay, what's really cool about this is like we can engineer proteins at the sequence level to get some specific structure, to get some particular function, right? And to all of us who are following the news or in this research space, whichever applies to you, like we all know that there's a revolution going on in protein design, right? There is kind of the idea of engineering sequences to get functions is just a way to say that can we engineer a protein sequence to encode for a particular structure? And the classical ways of doing this include screening, building a lot of variants, checking which one has the function you have, looking at evolutionary methods like the ones that Francis Arnold and others have popularized, coming up with more and more efficient ways to determine experimental structures, X-ray crystallography, cryo-EM methods, things like that. And all of that has given us many different proteins or understanding of how many different proteins work, but that's been substantially accelerated in the last 5 years because we now have a shortcut, right? We have a cheat code that allows us to go from sequence to structure. And this is essentially driving a new paradigm because we now have like a complicated neural network, not particularly interpretable, but at the end of the day, a large mathematical function that goes from the sequence of a particular protein and predicts what the accurate structure is, right? So it's accurate in the sense that it can match experimental resolution for the structures of proteins we know. It's cheap in that a structure prediction costs like 10 cents now instead of 10 to the power $4. So this is something you can screen on a computer to design stuff. And it's a mathematical function. So basically, if you have sequence and you have some complex F of sequence which predicts the structure, if you want to do the design process, kind of doing F inverse, right? Like if you can do F inverse of structure target, you can now identify new sequences that build structures that you care about. In practice, doing something like this involves computing something like TF over ES, right? You have to be able to understand how structure changes with sequence so that you can invert and get results. It's remarkable. Okay, this was just introduction. It's really fascinating to me because now we can go invert these neural networks. There's lots of methods people do this through just direct inversion, inversion that's aided by these diffusion models, things like that, to build new sequences, including sequences that have never been seen in evolution that encode structure. Bill?
So what is the active?
Like, okay, so everything using a computational model is only helpful for an experimental outcome if it is a good emulator of the experiment. Things like AlphaFold for the proteins that we have tested and known on the protein data bank are essentially like matching experimental accuracy. Like that's the gold standard. Like if you can get as low as experimental accuracy, then you're good. There are precise numbers like what's the Armstrong resolution of a protein and things like that. But that's what accuracy means. Like, can you get as close as an experiment?
So on a new one.
On a completely new sequence, I think the jury is out. It depends on what new is. So there are people who have synthesized sequences that come from the same family, increasingly distant in sequence space. And there are a lot of success stories where they've then crystallized and found that it matches AlphaFold. There are parts of sequence space that have never been seen by AlphaFold just because sequence space is infinitely large. And I think that it's less clear whether this is a interpolator or a generalized function that truly understands the mathematical landscape of sequence structure. We don't know the answer. Yeah. Alistair?
How one-to-one is this?
Oh, it is certainly not one-to-one. I just wrote it like this out of ease of words. Like it's not formally invertible in the mathematical sense. And that's something you exploit, right? Because you know many different sequences can encode for the same structure. So you want to find new sequences. So that allows you to find different parts of sequence space. It's also formally true that a sequence can encode different structures with different probabilities, and that's what we're going to talk about today. They do it in the mathematical sense that like there are proteins that can exhibit two different stable structures and flip between them. But the generalization of this is a sequence doesn't occupy a particular structure. If you're a statistical physicist, you'd say there's a free energy landscape and that every structure has its own probability that depends on the ensemble it's derived from, right? So it lives in a world of conformations. Speaking of living in a world of conformations, this is kind of important to think about because structure is a good starting point, but it's not enough. This is a simulation from D.E. Shaw of a molecular clasp, like this protein binds its target. There's no way you can design this purely from structure, right? This happens through some seemingly rare event where the arms of this protein come out and catch this protein. Okay, but there's an even more extreme case where structure is not all you care about. And that's what's known as a disordered region. Okay, so anyone knows about what these are? These are parts of proteins that are unimaginatively disordered. What that means is that they lack any stable secondary, tertiary, quaternary structures. They behave like a polymer that's fluctuating in solution. And this disordered protein is not an exotic beast that is unusual to particular organisms. 1/3 of all the proteins in the human proteome are disordered. Right? So if AlphaFold solves 67% of the problem, 1/3 still remains to be solved. And this is like an all-atom simulation of this. The point of this is that to understand function of proteins like this, it's not going from a sequence to a static structure. It is going from a sequence to a movie like that to understand what are the conformations these proteins occupy, and then how do they encode for properties like how it binds, things like that.
Does AlphaFold tell you if something's intrinsically disordered too?
Yeah, so here's, do I have it on the next slide? No, I don't. Let me get to that actually right now. Okay, so if I go to AlphaFold and say, can you predict the structure of a protein? This is tubulin, it's fully structured. It'll give me something that looks like that. The fact that it's colored in blue means that AlphaFold thinks that this is what it should match an experiment. You overlay an experimental structure, it's bang on. You give a protein with disordered regions, it just predicts these random loops. And these loops are colored yellow or orange. That usually is AlphaFold's predicted certainty in like what the structure of that sequence is. It doesn't know what's going on. It says, I'm not sure that this is the structure, but I think it's going to be a loop. So AlphaFold is amongst the most reliable ways to identify whether a protein's domain is structured or not. Because if AlphaFold is not sure it's structured, it's most likely it's an intrinsically disordered protein. Todd, does that help? Okay.
Usually proteins have some, they're not like all the same, all of them they're disordered, right? They have structured domains as well as some parts like that.
Right. So a typical protein is not just intrinsically disordered. So only 5% of the human proteome completely lacks any structured domain. Most proteins have these kind of structured domains, the coil domain, some NOPs binding domain for another protein-protein interaction domain, and then there are IDR loops that are in between. So like how these domains work together are probably important in understanding their function, not any particular piece. Question?
So, for example, multiple metastatic structures where one of the conformations is maybe particularly rare, how does the scaling of like the computational expense of learning to like train the model to accurately predict that rare conformation compare with the scaling of doing like direct simulation?
Yeah, I mean, so that's a great question. And I don't have a simple answer for that. The question is like, if you have proteins, for example, the simplest example is if I have a protein that adopts two different conformations, one is exponentially rarer than the other, then like, can I just predict that? First, AlphaFold might not be able to predict it. To predict it, I might need to do a simulation, identify these proteins, and then figure out, can I train them on that? And I think we don't have a gold standard for this. For proteins that have alternating structures that are equally possible, there are clever tricks you can use because the landscape learned by AlphaFold can be sampled by doing mutation to get alternate structures. But at the end of the day, the gold standard is like it's a simulation in this case, or an experiment, but a simulation can capture everything. And then emulators of a simulation are a machine learning model. That their job is to say, can I approximate a simulation but faster? And I think kind of the job of an emulator is that it's faster, cheaper, but an approximation. And I think so today, I'm not going to talk about these two structure class proteins. I'm instead going to talk about intrinsically disordered protein regions, which instead occupy essentially an infinite ensemble of structures, but each with its own probability.
An idea. So I think about the entire system of proteins. These intrinsically disordered regions are what? They're some kind of helping or hurting efficiency or something like this.
Efficiency of what? I'm sorry. Are you asking perhaps? Good. So yeah, the question is like, what do they do? Are they just like spandrels of evolution hanging around and like, or are they actually more useful? Okay. Okay, so we know one huge important task, and this has been studied a lot in various organisms, fungi, bacteria, plants, animals, is that this flexibility in taking different sizes is actually exploited by cells to build sensors. So for example, if an IDP sits either on its own or between two domains, depending on whether it's folded on itself, it can make the overall protein in an auto-inhibited or a active state. And by the virtue of, for those who study polymers, they know that these kind of collapse transitions can be quite sharp. So IDPs have evolved to like exploit these kind of collapse transitions for regulating function. And because this is made of amino acids, they're sensitive to biological signals like phosphorylation, methylation. But also these collapse transitions depend on the underlying physical chemistry. So the salt concentration, the overall excluded volume, crowding, temperature. So basically a lot of IDPs are temperature sensors, salt sensors, pH sensors. They respond in a phosphorylation dependent manner to activate or auto-inhibit a protein. So that's one class of tasks they do. There's more. I don't think I'll go through all of them. They can also just bind, like protein-protein interactions are thought to be the basis of functions. IDPs can bind without adopting any secondary structure and still provide specificity. Question?
Can you comment on the notion which I saw that the disordered regions are responsible for promiscuous binding between proteins because they are not folded already, so they can fold in response to what they encounter?
Yeah. So so promiscuous often is used to imply non-selective. So I think perhaps that's not what you meant.
I meant the many part.
Yes. So IDPs definitely have a role in that they can bind to multiple different targets. And one of the key things IDPs can do is that they can fold upon binding. I think that's an example you're pointing out. So for example, in transcription, many of the co-activators that sit might need to respond to different transcription factor signals. And the same IDP, because it's plastic and changing, can have a way to respond to different structures and still integrate that signal. So molecular information processing, if you want to call it that, is something that's really aided. You're not like forced into a particular structure and kind of like limited to that.
Are there any specific like biological processes where these IDPs are more prominent, where they're playing a more prominent role? Or is all pretty much all?
This kind of is an opinionated answer in the sense that people have their own perceptions. I would say that they are known in binding, they are known in structural, just like cell wall building, there are IDP kind of structural motifs, especially for these things called condensates that are compartments that form inside cell. I would say that they are perhaps a little bit underrepresented in catalysis, in doing things like transformations of small molecules and running the metabolic processes, and perhaps a bit overrepresented in sensors. Of course, they're very rarely on the membrane themselves because you need to fold to go through the membrane, but they are attached to membrane proteins. Todd?
You mentioned molecular simulation as being like a gold standard, but is the accuracy, like the force field, is that even as good as if your goal for molecular simulation was structure prediction, not just dynamics? Does the accuracy even reach like AlphaFold type accuracy?
Yeah, so here's the interesting part, right? Like if you take a structured protein and if you simulate it on a computer, it's pretty bad.
Yeah.
All the state-of-the-art force fields suck.
That's what I would have expected.
Yes, that's absolutely true. I mean, they suck to different extents, so I shouldn't be too harsh, but there's a degree in which they're all more or less not representative of experiment. This is not true for IDPs, however, because a lot of coarse-grained methods have emerged in the last few years. This is not to say they're amazing, but they usually very reliably predict ensemble properties benchmarked by NMR experiments and things like that. So they are pretty good.
Yeah, I mean, that's a different level of like prediction than if you're saying, hey, I want to know what is the RMSD with a particular structure, which is what people are often trying to do.
Yeah, so it's aided in the part because the function of an IDP is cannot be prescribed down to how a particular stable structure looks. So instead, people, what they care about is like, I have this fluctuating polymer in solution, can I get some ensemble statistics? Can I get the distribution of sizes it takes? And those things are captured in experiment and matched by simulation. And so the goal of these simulations are usually to just predict those ensemble properties. Certainly, it's not like what it is for structured proteins where force fields, even coarse-grained force fields, pretty much still continue to not do the predictive task. Okay. Okay, so maybe I'll summarize all of that by saying like the logic here for these kinds of proteins are often sequence encodes for a large ensemble. Then there's some ensemble average property that's important for their function. You know, as linkers that flexibly respond, as sensors, as building these condensets. Okay. Right, how do we design these things? Because we don't have these simple invertible f of s to go. This goes to Todd's question. Right now, the gold standard is actually coarse-grained simulations. There are multiple techniques. They're all not bulletproof, but they're pretty good at matching experiments, which is the standard for a molecular simulation. So you can take a sequence, put it in a computer, simulate it, compare it with multimodal data of measuring its hydrodynamic radius, its entire scattering function from NMR, and these things map reasonably well. So the f of s here is not a function, but it's an MD simulation, right? And an MD simulation is nothing fancy. It's just following Newton's laws for a long time. Question.
How coarse-grained is it? Is it like by math level? Or...
Yeah, so these are usually coarse-grained to one amino acid being one bead. There are more and less coarse-grained approaches. There are ones that are implicit, all atom fields. They actually are less reliable because they follow the traditional kind of the amber charm logic, and they just fail. So usually when I'm talking about coarse-grained, I mean one amino acid is equal to one bead. They are all implicit solvent. So they've all been trained to reproduce properties across a range of salt temperature conditions that are typical, 150 millimolar salt temperatures that are within, tens of Kelvin of 300 Kelvin. Okay, more questions? Okay, so the goal here is our approach is, a little straightforward when I say it like this, is that an MD simulation sounds like a fancy thing, but it's just following Newton's law over time. There's no analytical expression, otherwise you wouldn't do an MD simulation. So you just have to solve this many-body problem. But in principle, this is also mathematically well-defined. So our goal is going to be asking, can we just basically take a simulation that looks like that of a particular sequence, and then rationally invert the simulation, compute a gradient through the simulation, so computing DMD simulation over, or D properties from MD simulation over T sequence to move forward. Okay, that's going to be the approach. It's not straightforward. So I'll walk through a couple of minutes of how we set this up. Okay, I have time. Okay, questions. So we already discussed what the idea is. For, say, a structure problem, you have sequence, neural network, which is a proxy for a fancy function. Structure, you have a target structure. And if you want to, so let me just write this down here. Sequence, structure, you have a good predictor, that's f of s. You have a target structure, and you just want to identify a sequence that gets you to the target structure. You usually would do something like minimize a loss. That's like that. And then structure is a function of sequence. So you just need to compute the minima of this quantity. So you just need to be able to get the structure of sequence divided by D sequence, which is whatever, taking backprop and getting gradients through this neural network. Moving forward, if it's a more complicated function, you just have to figure out how to do this part. Okay, so the idea is we are just going to write MD completely differentiably. So right now you can't take lamps and take a gradient through it because it doesn't plug and play with all the nice machine learning architecture that's been developed to compute gradients through complex programs. This is the state of the art, right? Today you can write a computer program, an arbitrary composable computer program, and just take the gradient of the computer program entirely using these differentiable programming libraries. And what we're going to do is just write the molecular dynamics simulation. Actually, we are not. Google has spent significant amount of time writing this. So we just like hang with them and figure out how to tweak it to our problem to basically use a differentiable molecular dynamics simulation. So then it's super simple. We have sequence differentiable molecular dynamics simulation property. We just need to take a gradient through. Okay. Should that work? Question.
Really computationally intensive and slow. So how fast can you auto differentiate through it?
Great question. So maybe that's the next slide. Okay. So so in concept, this idea sounds great, but like there's lots of structural, pun intended, problems, including the one that was raised right now. Like the goal is to say that we want to go, we have some parameters. In our case, it's a sequence. We have some property, and we just want to diff through MD. But MD is long. Like usually you have to run, several million, tens of millions of steps to even get ergodic equilibrium sampling. And that's 10 million different mathematical computations as opposed to 100 layers of a neural network or 1,000 layers. Like how do you actually get past the memory constraints that come with it? So let me, and there's one more thing, of course. Sequences are discrete. Biology doesn't work with continuous sequences. And usually when you write down a gradient, it's only infinitesimally true. So you have to be able to move sequence very, very small ways, which you can't do if you're working with discrete sequence. So there are two challenges. How do you make this continuous? And how do you actually like solve the harder challenge? Like how do you actually take the gradient? Okay, so for sequences, because almost all of these interactions depend on pair potentials, we're just going to do something fairly straightforward. We will take a discrete sequence and relax it into a continuous probability. So this is just now, our design parameter is not the sequence, it's a matrix which represents what's the probability a particular location is an amino acid J. And then we just compute the pair potential as a matrix vector product between any pairs of species. And if this matrix had one hot columns, or how did I write it down? One hot rows, then it's just a discrete sequence, right? You can just read out the sequence if it's only ones and zeros. But other than that, if it's a probability matrix, it's a continuous representation. So eventually, when we finish optimizing, we want to guide this matrix to become discrete. So we'll just do that by annealing it. There are lots of standard techniques that are there. That's the easy part. The hard part is if I take gradients through a simulation, so the x-axis here is the length of a simulation, so how many steps you take. And the y-axis is in log scale, the size of the gradient of, in this case, say the RG, the radius of gyration over the simulation step. You can see it blows up. And this is because to compute this backprop, you need to basically do D property over D sequence. You have to do chain rule, which means you have to like basically do the chain rule through 50,000 MD steps. And if, these things explode if the underlying Jacobian is not stable. So almost certainly in all cases, they will explode. So if you want the gradients not to blow up, you run very small simulations. If you run very small simulations, are you even capturing equilibrium properties? So if you instead do straight through optimization, so here's the case where we want to get to an RG that's a particular number, we start there. If you just do small simulations so that your gradient doesn't blow up, the gradient information is actually not useful. So you don't actually do any optimization. Okay, so the conceptual thing I wrote down cannot be done for the reasons asked. You cannot actually optimize over an entire MD simulation for this problem because you need to sample long enough to get there. So what do? We use a very, very nice approach that was developed by Thaler and Zavarlab and independently by Zhang. It's called trajectory reweighting. So let me just outline the idea. The idea is that we usually go from sequence and force field through a simulator. So in this case, molecular dynamics, but in principle, any arbitrary simulator to get a bunch of equilibrium states. And then from those states, we compute the ensemble average property of interest, how well it binds, how well it folds, what's the radius of gyration, things like that. And in principle, what we want to take is del theta of the observable value. So if there's a way to short circuit going through the simulator, that's going to solve all our problems, because all we can do is independently gather the states and then just backprop through an expression for the weighted observable through the sequence. Okay, Todd.
It's a fancy way of saying that you've had a temporal average for a long time.
No averaging is
And
happening.
instead, you're gonna now have a bunch of copies that do short time evolution.
Not at all what I'm saying. So it's not what.
Why would you not do that?
Yeah, because the point is the gradient blows up even in the decorrelation time of that to work, right? You're saying I need to batch sufficiently large and short trajectories, and I can still get that information. The gradient will still blow up in those sufficiently small batches. Actually, just backpropping through MD is really hard even for like very short time scales. What I'm saying is, like, basically the point of MD is that we are going to simulate this, collect snapshots and compute a property from that. So if you just acted as if some agent gave you those snapshots, whether it came from an MD simulation or a Monte Carlo method or your favorite like ChatGPT, Sora model, whatever it is, in principle, just having these snapshots should be enough to take the gradient if it's at equilibrium. So that's something we know. So actually, let me walk through this a little bit more, why this is an important idea. Right, so if you have a sequence and you run a simulation, collect a bunch of snapshots, right? So you have 5 snapshots. And then I want to say, how do I compute? And then you measure a property, right? Like when you average, what you typically do in a simulation is you take the snapshots and compute the average. That's like just doing O1 plus O2 plus O3 plus O4 plus O5 divided by 5. Okay, and if I want to compute the gradient of O with respect to sequence, like if you actually do this in a simulation, it's just a bunch of snapshots. There's no explicit dependence on this property on any of your simulation parameters. So if you compute an explicit gradient, you'll just get 0 because you just collected a bunch of snapshots and tried to compute a derivative. But there's this perturbative trick that actually was developed in liquid state theory many eons ago. But the idea was that you can compute the average not just as a sum of snapshots divided by n, but approximately as a sum of weights for each snapshot. But this weight comes from a probability function that we know because we know physics. So in particular, we write down that this weight depends on p theta. So this is the sequence parameter. So let me call it ps of OI by p sequence hat of OI. So it's asking, as I move the sequence a little bit, what is the probability of that particular conformation change? And we know that for this, it's exponential minus beta u if it's the canonical ensemble and you're at equilibrium. And obviously, if your reference ensemble and the ensemble that you care about are identical, this just becomes 1, and you sum down by the number of states. So this is just 1 over n, which is how you compute an average O1 plus O2 plus O3 divided by number of snapshots. But by using this function, this is now explicitly a function of this, which is a function of S. So you know how your individual snapshots depend on the sequence, so you can compute this gradient accurately. What it means is that, so that's one thing that it solves. It means that you can actually take this gradient efficiently, quickly, and independent of having the entire simulator itself being differentiable. You can literally just take the sequence, throw it into LAMPS or whatever non-differentiable code you want, collect snapshots, but exploit the fact that you know you're near equilibrium to do this. There's another thing you can do, which is you can reuse reference states. Like if your sequence is moving only slowly, then you can assume that the reference ensemble, these collection of states that you have sampled, can more or less describe what you get from the new sequence. And you only have to re-simulate when the set of states that you collect no longer match the underlying reference ensemble, which you can compute through some statistical property by the entropy of the ensemble itself. Does that make sense? Too fast? No. Okay, so this sidesteps. I think what I'm trying to say and underselling is that the idea of this is generally true when you go from any parameter through a model to collect some properties, as long as you know what the underlying physics of the equilibrium distribution should be. So it can come from an MD, it can come from an MC, it can be unrelated to this problem. In general, we can take this approach. It was developed by Thaler and Zavadlav to actually initially fine-tune neural network potentials to compute those gradients. Okay.
I need to show you, but I've seen that formula, but it was from 1997.
The first time this kind of thing that I've seen comes from the 60s, where what people wanted to do were take a hot sphere fluid, which Baxter and others have studied. I guess Todd might know this stuff. I don't know the other people. Folks might know this, but if you want to look at liquids that have interactions, it's a little hard to solve their statistical mechanics. There's lots of approaches, but one approach people used to take is collect reference states as predicted from the hot sphere ensemble, and then apply this perturbative trick, saying that you just have like a small deviation in your interactions that's mildly attractive, and then compute what your ensemble average is. It's not used to compute a gradient, it's just used to compute an ensemble average property, like the pressure or things like that. And it's pretty useful trick. It's just a variational trick.
So you're talking about an important sample.
Yeah.
Yes.
But there's a degree to which this is not just a generic important sampling, this is an important sampling with a small perturbative.
Yes. It's only true in the small perturbative limit. And basically something like computing Wi log Wi is going to tell you, like, are you still close enough? And if that's not large enough, then you have to resample and then recompute. So in practice, what it looks like is an optimization will run an MD simulation. We can maybe go a couple of steps by computing the gradients to the old samples, but already we now have to redo the important sampling to get the states again. And so we keep moving this.
Yeah, so I know it is something called the balance heuristic.
The balance heuristic.
It was devised for Monte Carlo simulation, important sample Monte Carlo simulations in computer graphics.
Okay, cool. I should learn more. I don't know that. Okay, but the idea is like this kind of perturbative small approach gives you an explicit formula. You can take a gradient over a simulation. So we can do this optimization that I talked about earlier. This is no longer a pipe dream. You can formally do this optimization. Okay, we have a few more minutes. The rest of the talk is like results. We can still go over them if you want, but like this is the important stuff. Okay, so maybe I'll talk about a design problem. Okay, so a design problem is you have a polymer, it's fluctuating an IDP, and it's going to occupy an ensemble of states with an effective hydrodynamic radius or radius of gyration. And for those who are interested by these kinds of things, these are important for how well an IDP behaves, like an efficient linker between two domains, or what its size is within the system. And the question is, can we design a protein with a target RG, right? So we start off with this probability distribution that's uniform. So you can take any sequence it wants and eventually do gradient descent. The one thing I didn't mention is if I take a matrix like this, it's just shown in this logo form, and I just do gradient descent, I can still end up with a continuous sequence at the end. There is nothing called a continuous sequence in biology. So we add a little bit of a driving force to anneal this towards a discrete sequence. And what that means, you're just seeing this again, is that you plot something like a probability distribution. By the end of the simulation, you want each site to just have one residue. So you just want it to be one column. So you start the simulation, compute the gradients, do your optimization. And I like movies. So like eventually you converge, kind of looks almost annealed. We then take the annealed sequence. You can basically take this and say, what is the first sequence that comes, simulate that and validate whether your prediction works. It's bang on target. Okay. What's important is that this is not like a one-off trick. Here's like optimization that's done over an entire range of RG values that is permissible for a sequence of a finite size. So we can design sequences to reach any of this. Because it's a probabilistic sampling method, you can literally rerun the simulation with a different seed and get a new sequence. So sequence design becomes just a matter of sampling enough. And I think it's not maybe as clear from the trajectory reweighting trick, but it's not tied to any particular force field or any particular physics model. Like we can swap out the force field and just use the same concept and then do the same design problem all over again. Okay. I'll skip this. Another complementary approach, actually I'll just say quickly, a complementary approach that people are adopting is doing something that was brought up in the discussion before. People run a lot of MD simulations, measure properties, and just train a neural network to predict the property from the sequence. It's cheaper that way, right? So this is an example of this property. It's called Albatross. It's a machine learning predictor that's been trained on tens of thousands of simulation data to say that if I give you a sequence, can you predict what its radius of gyration is at some standard typical condition? And one of the advantages of optimizing over a simulation rather than an approximation of a simulation is that you can actually discover solutions. So this is an example of a very stretched polymer that approximations can't work for, right? Like approximations are by construction approximations, and they are only as good as the data set they generalize over. So if you want to find solutions outside of that, inverting a simulation gives you access to data points that are not there. It also teaches you in principle how to improve your machine learning models by sampling simulations that are at the far edges of parameter space. Okay, I'm going to skip this. This is interesting. It's another design problem about loops and linkers. But I'd say one of the things we're most excited about is designing sensors. So because an physics-based simulation like an MD simulation can extend to different regimes by changing your physical parameters like temperature, salt, modeling their dependence appropriately, how the charges or the residues change. One of the things we were interested in is asking, can we build synthetic disordered proteins that will respond to a stimuli either as a contractor or an expander? Okay, so that's how it changes. That's what the design criteria is. And we actually learned, I would say, unintuitive to us and unintuitive to our friends who design these IDPs solutions. So for example, if you want a sequence to contract when you add salt, the primary addition of adding salt is to screen charge interactions. So you might say that I build an extremely positively charged residue, and if I add salt, that should contract a bit. But our solution finds that you actually need to add these little Velcro stickers that are scattered around the sequence so that when the salt goes up and the positive charges stop repelling each other, these Velcros will come in and zip. And so you get a really, really amplified contractor. This FX size, I guess these numbers are kind of meaningless to you. This is kind of like almost the entire dynamic range of a self-avoiding random walk. Like it goes from expanded to like a compact polymer. Even more, like if you want to design an expander, so this is something we actually asked some of our experimental friends. They said, here's a simple logic. You build a swath of positively charged residues and then a swath of negatively charged residues. So this polymer likes to bind to itself. But when you add salt, those interactions weaken, so it'll split out and become a contract. So we thought, okay, we'll optimize for an expander and see whether we can learn this solution, which our in situ experts thought would be like the best. So actually you can simulate this, and that doesn't work really well. It's like a 10% change. But the solution that we discover is actually a positive, which is this blue thing, and a negatively charged loop, but they have a linker in between. And this linker in between is entropic. So at low salt, the interactions are strong enough that when they come and fold upon themselves, they can stick. There's an entropic barrier they can overcome. But when you have high salt, this entropic barrier is just large enough to prevent this collapse, which means you have this huge amplification of just a binding unbinding event within a molecule. And you can notice here the FX size is again like almost twofold, which is substantially large. Okay, I am very close to time, so
I have questions.
particular.
You always need like an IDP to make a sensor. I mean, would it not be like a already defined unique structure methods.
You can do whatever you want, but the point of this was to highlight that you can also do it through IDPs. This is not discounting the fact that structured proteins can behave as sensors.
I'm just trying to see on the long term, not for basic science purposes, but for engineering purposes as well.
Well, for engineering purposes, actually, this is really useful because so we have one of your colleagues, Will Sneed, who's a friend of mine and collaborator, is actually trying to get these things purified and tested. And the reason they're excited in using IDPs for sensors is that you can get these kind of salt amplifiers pretty readily. For structured domains, you have to discover proteins that have done this in the first place and then try to build an evolutionary homologue. And I'm sure there are examples. These are just different strategies. They're easy to keep in solution often. When they're expanded, they're soluble. There's some design reasons they're easier to build too. Yeah, I'm not sure there's something like innately better about it. It's just a design space that's not been explored previously. Yeah. Okay, and maybe last but not least, I'll jump over this too. Like basically another thing AlphaFold does is it does binder design. I'm sorry? Okay, I'll do a couple of minutes more then. So one of the things AlphaFold is really good at is finding how two proteins bind. And so people have developed all these awesome mini binders that can go and bind a protein of target. There are also proteins that are completely disordered and bind and have picomolar affinity. Okay, so picomolar affinity for the biologists in the room is non-biologists in the room is incredible. Like it's incredibly strong. It's amongst the strongest interactions known in biology. So the question is like, can we actually design binders to a disordered protein that has strong binding coefficient despite lacking structure? And this is interesting because there's a lot of disordered proteins that are mutated in disease. And so the question is, can we design peptides that go bind them, sequester them? I'll skip the result. You can do this in principle. There's lots of interesting features in these optimizations. Like as we run the optimization in the x-axis, and we're measuring sort of how close the bound and unbound structure are. Like in our optimization, we just like have this somewhat sudden transition between like sampling sequences that come from an ensemble that don't bind at all to suddenly being bound. So these binding events are like, we think it represents an underlying cooperativity in the binding events themselves. Like as soon as we get to strong enough bound that's measured within our simulation parameters, we just lock into a binder and then just anneal to a particular sequence. Okay, there's a paper under review. If you want to learn more, all the details are there. And then I'll just give you 2 minutes of what we are doing now. So there's lots more to do here. The parameters we have been inverting are sequence for sequence design, but every MD simulation, there's a sequence plus the force fields you put in there. The same logic can be used to say, how do I improve force fields to match experiments better, right? Because we take a gradient with respect to force field instead of sequence. So we take a collection of experiments where people have measured an experimental property and ask, how do we update our existing force fields, which is usually done in a very ad hoc way, right? For those who know about force field development, it's a pretty artisanal kind of cottage industry. So this is like potentially now a rational way to like fine tune force fields. Depending on the experiment too, you could have like a binding force field, you could have a wider temperature range force field. The other thing we're doing is that at the end of the day, given everything, molecular dynamics is still a relatively expensive calculation. It just happens to be a good calculation that matches experiments, so we use it. So one of the things we've been thinking about is, can we short circuit by replacing with a pretty accurate physics model? Doesn't have to be perfectly accurate. And then can we build heuristics to then test back via simulation that you can build this? Okay, so we've been using approaches from statistical physics that helps us calculate the virial coefficient of these molecules in particular. Okay, so the idea is that instead of running a simulation differentiably, you do the theory calculation differentiably. It's not particularly profound. And then we go and ask, can we design problems? So this is what I told you before, a salt contractor. We just went over the example. Can we do the same thing, but using a theory now instead of a simulation? And we basically can uncover sequences that do this. You see effect sizes that are large. and you see the same kind of solution structurally, positively charged residues with these little Velcros. In between, the most important thing is this theory calculation took like basically a few seconds instead of 3 hours on a A100 GPU. So a few seconds on a MacBook Pro, as opposed to running 3 hours on an A100 80 GB GPU to run multiple MD simulations and invert that. So it's actually faster. But because it's faster, you can ask new kinds of design questions. Because these are conformationally plastic ensembles, can we start building these molecular information processing gates, right? Like, can you take a protein that only when it's phosphorylated and at low salt will remain expanded? It's kind of like an AND gate or an ON gate, but in other cases will remain compact, and we can like uncover sequences that effectively do that. We're also now going to ask, can we design sequences that can form, either well-mixed state that are dissolved, form phases that recruit specific molecules, form multi-phase assemblies using these kinds of theories. Okay, now I will formally stop after having threatened it multiple times. Yeah. Questions?
I want to come back to my earlier question in the light of what you have shown in the last couple of slides. You are designing binders, meaning that you are designing, I didn't figure out, is it just a short sequence or a full-blown IDP, IDR, which binds another IDR? So can you do this kind of n by n matrix of IDRs, which will have a specific binding properties?
Yeah. It's first great question. I'll tell you what we do. We design an IDP that binds to a target IDP. So everything is an IDP. And that's in large part because these molecular models for describing folded proteins are much less reliable. And we've only done 2 test cases, which is like, can we find a peptide of a finite length, 30 amino acids or so, that's disordered and binds to proteins, disordered domains of like prominent IDPs like FUS and V3. These also just happen to be the IDPs that my friend works on. So we purify and test things eventually. So it's motivated by experimental connections. We can do exactly what you're saying. It's a lot of GPU time to burn. But
1,000 times less than would have if you were doing molecular dynamics simulation.
No. So the binders were done with the molecular dynamics. Now, if we can do this with physics directly without simulation, that's where we're going.
I see.
We are actually, virial coefficients roughly predict things like affinity. They're not exact. So we're trying to see whether we can design virial coefficient matrices n by m that look like a particular target thing. So they predict kind of like how effective interactions are between 2 species. They're not really like an affinity. They're getting there very slowly, but getting there.
Might be important for dimerization networks which are being used for computing. So that is something we can talk after.
Yes, please. Yeah, also I think there's this general question. Of course, it's only as good as these force fields are a model of experiment. But this general question of like how much specificity can you stuff into an IDP, right? Like, can you only, like, is it promiscuous and non-selective, or can it be promiscuous and selective? Can you actually build 10 IDPs that can mutually bind only their one preferred partner? I think this is somewhat an open question. So it'll be interesting to see it.
What physical regimes does your procedure tend to be more expensive? Not necessarily with respect to like the number of like particles or amino acids per se, but like maybe characteristics of the dynamics or the ergodicity or.
Yes, good question. So the primary expense, because we're using the reweighting trick, is not the gradient calculation. It's just computing the MD simulation. So anything that makes that harder is a problem. So if there's a system which takes longer to get to equilibrium, so longer sequences famously take longer time to reach equilibrium, there are ways to short circuit that by like initializing them with like pre-trained ensembles that are closer to equilibrium than just how we start. We start with a long chain that's like linear. It's a horrible way to start, but it's kind of like the way we start to emphasize that even with this case, we are able to discover. There's another limitation of this approach, which is like sometimes what you're interested in is not an equilibrium property, many times. And like the whole reweighting trick assumes you know the probability of stuff, which maybe you know at equilibrium, maybe you can make up something near equilibrium using like linear response or things like that. But like if you truly want non-equilibrium properties, this approach doesn't work. You can take like a reinforcement steering kind of approach to push simulations through. It's still expensive. Yeah.
Is the equilibrium mixing, for example, of like a salt solution? So when you're working with sensors, you have to account for like a change in not just like the local detailed balance of the conformational space, but like of the solution space. Does that ever become
Implicit solvent and ions. So all that stuff is lost. So if you build more details into your model, it takes longer. So if you want to figure out how salt ion entropy changes, which is a really important factor, it improves. So basically anything that makes your forward calculation more expensive makes this harder. And in some sense, the first proof of example we showed were the ones where that forward calculation was not obscene. Like we still can't invert. I think you've seen these simulations. Like studying the face behavior, it's a really expensive calculation to run. Like one calculation takes several hours. So like, we are not at Google or Microsoft. We can't just like burn GPUs time forever. So some short circuit is going to be important. It's also useful generally to learn like how well you can short circuit complex things with simple physics. Yeah.